After leaching and physical fragmentation, decomposition primarily involves chemical alteration. This is done by a host of different organisms. Fungi and bacteria are the main initial decomposers of terrestrial dead plant material. They account for about 95% of the total decomposer biomass in respiration. So here in this section, we're really going to focus in on some of the main organisms, the fungi and the bacteria, that account for most of the decomposition of organic matter. This is figure 7.2, which gives you an idea of the size of the organisms that are associated with decomposition. So we generally talk about microflora or microfauna, and flora is just a general term which involves microbes. But the microbial community is small, often less than 100 micrometers. Bacteria are the smallest, and fungi have a range of sizes that they are. What's unique about fungi is that they have hyphae. And hyphae are strands of fungi that are connected to one another. And by being connected and sharing resources, they can move resources over relatively large distances and act as an integrated organism instead of just being a small individual cell. These hyphae are important because they can import resources to the site of decomposition. So nitrogen or carbon, for example, that's necessary for growing new material, new biomass, can be imported from outside of the site of local decomposition. So they can integrate over much larger areas in their decomposition. Fungi also have complex enzyme systems. Basically, any different compound can be decomposed or degraded by the enzymes of fungi. In general, there are two types of fungi that are associated with decomposition. The first is saprotrophic. And the root word here for sapro means rotten. So it's fungi that eat rotten material. This is an example of a saprotrophic fungi, but there's two major classes that we'll talk about here. The first are called white rot fungi, and their unique contribution is the ability to degrade lignin. Lignin is a class of compounds that are very recalcitrant and hard to decompose, and white rot fungi specialize in their ability to decompose lignin. In addition to white rot fungi, there are brown rot fungi. And these also decompose lignin, but they leave one of the major groups associated with lignin, the phenols. And they're not quite as good at decomposing lignin completely as white rots are. These fungi are favored under low nitrogen concentrations, and they primarily mine the lignin not only for energy, but also for the nitrogen. And if you were to fertilize, for example, and add nitrogen, these tend to be outcompeted relatively quickly by other decomposers. And so when you're in systems that have low nitrogen availability, such as with wood decomposition, you tend to find white or brown rot fungi. Now, fungi have a number of different characteristics that are unique about them. And we talked about the complex enzyme systems that they have, but also they have high C to N ratios. And this in general means that when they produce their tissues, there's a lot of carbon and not a lot of nitrogen. And it's been interpreted that it means is that these organisms have a lower requirement for nitrogen and therefore would be found under situations where you have lower nitrogen availability, like in the wood systems. Now, in addition to saprotrophic fungi, you have mycorrhizal fungi. And mycorrhizal fungi are fungi that are symbiotic with plants. And that symbiosis is that plants provide the carbon for the mycorrhizal fungi. And the fungi then do the decomposition and transport a lot of the resultant nutrients that come out of decomposition into the plants. Now, in general, it's important to know that the fungi in soils are incredibly diverse. And it's estimated that somewhere between 10 to 100 times greater taxonomic richness in soils than there are plants in a given area. So there's a lot of different types of fungi in any one particular gram or handful of soil, and a lot more richness in taxonomy there than you'd find in the plants above. Now, fungi are one class of organisms that decompose organic matter. Bacteria and Archaea are the others. The small size and large surface to volume ratio of bacteria and archaea enable them to rapidly absorb soluble substrates and to grow and divide quickly in substrate-rich zones. So fungi we thought of as being in relatively low nutrient areas, and they have a relatively large size for microbes. They have hyphae, and they can move resources from one place to another. Bacteria and archaea, on the other hand, are relatively small, and we had seen that. And this gives them a large surface to volume ratio, which allows them to absorb nutrients and other compounds relatively well. And they tend to grow relatively quickly. And so we tend to find them in substrate-rich areas as opposed to fungi. So you can see individual microbes can grow in clusters, but they are really small compared to most of the plant parts that they would be decomposing, or the organic matter. And we think about bacteria and fungi, that these really are two of the three main evolutionary domains that there are, and they're very diverse. They tend to have higher resource requirements than fungi, and they also have a diverse set of functions that they can carry out. Bacteria and archaea tend also to dominate the rhizosphere, that area around roots that have a lot of resources. So a lot of carbon is going to be there, a lot of organic carbons, compounds, and a lot of nutrients too. So the rhizosphere tends to be more dominated by bacteria and archaea. Now, among the different types of bacteria, one of them that I'll talk about today are actinobacteria, and sometimes we'll just shorten that to actinos. And actinobacteria are the fungi of the bacteria. So they have individual cells, but form long strands when they link together. And so they're filamentous in that case, and to a certain degree are analogous to hyphae. In addition, they have some ability to degrade lignin, and so we find them in relatively resource-poor areas. Now, for a given gram of soil or handful of soil, there's a huge number of bacteria that are in there. And most of the bacteria that you find in soils are actually metabolically inactive. They may become active under certain circumstances, but as far as we know, most of those bacteria actually at one point in time are not going to be active metabolically.